Hey everyone, welcome back to Windows 8 Studies. I hope you all are doing well. So today we'll be learning about TAB sequencing. Till now we have learned about bisulfite sequencing, we have learned about oxidative bisulfite sequencing, and today we'll learn about a new method, and that is TAB sequencing. So the full form of TAB sequencing is Tet-assisted bisulfite sequencing. Okay, so this is a variant of bisulfite sequencing. This is a new version of it. So so before learning about TAP sequencing, let's do a quick recap of bisulfite sequencing and oxidative bisulfite sequencing. So in bisulfite sequencing, the cytosine is converted into uracil. The 5-methyl cytosine is read, it stays as it is, and that is why it is read as cytosine. This uracil is read as thymine, while 5-hydroxymethyl cytosine is also read as cytosine. Okay. In oxidative bisulfite sequencing, here in the bisulfite sequencing, you cannot differentiate between 5-methyl cytosine and 5-hydroxymethyl cytosine. But in oxidative bisulfite sequencing, you can differentiate between all those three: cytosine, the 5-methyl cytosine, and 5-hydroxymethyl cytosine. Why? Because cytosine is converted into uracil and it is read as thymine, and 5-methyl cytosine, it is not converted into, it is not deaminated, and that is why it is read as cytosine only. But this 5-hydroxymethyl cytosine, it is converted into 5-formyl cytosine, and then it is deaminated to uracil and read it as thymine. While in the TAP sequencing method, we are going to do bisulfite sequencing, but it will be Tet-assisted, means there will be involvement of Tet. There will be involvement of Tet enzymes, and Tet enzymes, we have learnt about this, they demethylate the DNA. Okay, so what do? They convert 5-methyl cytosine to all the way around 5-carboxyl cytosine. Okay, so they oxidize the 5-methyl cytosine and convert it into 5-carboxyl cytosine, on which TDG acts and it is converted to the cytosine. So that is the further process. So this is how it does the demethylation of DNA. So I hope you remember all this part. So in TAP sequencing also, just like the oxidative bisulfite sequencing, we will be able to differentiate between 5-methyl cytosine, 5-hydroxymethyl cytosine. Okay, both of these will be differentiated, and that is why we also say that TAP sequencing is helpful for detection of 5-hydroxymethyl cytosine. Why? So in the bisulfite sequencing, we can detect 5-methyl cytosine differentially from cytosine. In the oxidative bisulfite sequencing, we can detect 5-hydroxymethyl cytosine, and as well as in TAP sequencing also, we can detect the 5-hydroxymethyl cytosine. So let's learn this process step by step in details. So let's assume this is a DNA sequence. Okay, just let's take it here. Okay, let me just give me a second. So let's assume this is a DNA sequence. Okay, so I'll be drawing three types of the CpG dinucleotides. This is the unmethylated one, so it will have cytosine. This is CpG, this is the representation of CpG dinucleotide. I hope you all know that. So the shaded one is the 5-methyl cytosine. This green one, this is 5-hydroxymethyl cytosine. Okay, so those are the CpG dinucleotides. Okay. So let's assume that they are arranged somewhat like this on the DNA. Okay, let's assume this is 5-hydroxymethyl cytosine, and those are 5-methyl cytosines. So this is the cytosine, 5-methyl cytosine, 5-hydroxymethyl cytosine. This is 5-methyl cytosine, and this is the cytosine. Okay, so this is the DNA sequence. Okay, so first in the TAP sequencing, first we treat this DNA with an enzyme known as beta glucosyl transferase. So beta GT, beta glucosyl transferase. This enzyme glucosylates 5-hydroxy methyl Okay, so we need as with beta glucosyl transferase, we need UDP glucose. Okay, so UDP is uridine diphosphate glucose. Okay, so this is uridine diphosphate glucose. So this is the glucose provider. This is from this UDP glucose. We take the glucose group, and that is transferred to the 5-hydroxymethylcytosine, and this beta GT converts 5-hydroxymethylcytosine to 5-glucosyl hydroxymethylcytosine. Okay, so this glucosylates the hydroxymethylcytosine. So what happens that after the beta GT treatment, so let's just copy this DNA. Okay. So so what will happen that this 5-hydroxymethylcytosine, it will be glucosylated and it will be protected. So this got converted into 5-glucosyl hydroxymethylcytosine. Okay, so this happens due to the beta GT. After that, this is the first step of the TAB sequencing. After the beta GT treatment, we treat this DNA with Tet enzymes. Okay, so what will Tet enzymes do? Tet enzymes will convert 5-methylcytosine to 5-carboxyl cytosine. It can convert 5-hydroxymethylcytosine to all way around 5-carboxyl cytosine. Okay, but what will happen that here all of the 5 hydroxymethylcytosine, they are glucosylated, and that is why on this Tet enzyme cannot work. Okay, so Tet cannot work here on the 5-glucosyl hydroxymethyl cytosine. So what will be the result of this Tet incubation? So let's just copy it again. Okay, so just a second. So this will be the result will be that. Okay, so what will happen? This so the cytosine will stay as it is, because on the cytosine state enzyme cannot work. Those 5-methyl cytosine, they will be converted into 5-carboxyl cytosine. Okay, so this was protected with, this is 5-glucosyl hydroxymethyl cytosine. Okay, so this stays as it is. The cytosines stay as it is, but 5-methyl cytosine, they get converted into 5-carboxyl cytosines. Okay, so those pink ones are 5-carboxyl cytosine. Okay, so this will be the result. Okay, so this will be the formed. Now the third step in the TAB bisulfite sequencing will be, so it is Tet-assisted bisulfite sequencing. So you can guess this, what will be the next step. Okay, so the next step will be bisulfite treatment. Okay, so bisulfite treatment, here we treat it with sodium bisulfite. So sodium bisulfite, it works on 5 no, it works, just a second, it deaminates cytosine, converts it into uracil. It converts 5 cytosine to uracil, as well as 5-carboxyl cytosine to uracil. Okay, so what will happen that this on this DNA, the cytosines, it will be converted into uracil. So let's have a different color for it. So this is uracil. So 5-carboxyl cytosine, they will also be converted into uracil. Okay, So cytosine will be converted into uracil. 5-carboxyl cytosine, they will also be converted into uracils. These are uracil, uracil. The 5-hydroxymethyl cytosine, as it is protected, it will be, so this is 5-glucosyl hydroxymethyl cytosine. It will stay as it is. And again, this cytosine will be converted into uracil. Okay. So this will be the result. This will be the final DNA. And after that, we do sequencing. Okay, and in the sequencing, what will be the read? The read will show as thymine, thymine, cytosine, thymine, and thymine. Okay, so this is the detailed step-by-step procedure of TAB sequencing. Let's summarize it here. So, in what happens in TAB sequencing, the cytosine is converted to uracil and it is read as thymine. Why? Because it on it the bisulfite can act. Okay. The 5-methyl cytosine, it gets on this the trait works and it gets converted into 5-carboxyl cytosine, and this 5-carboxyl cytosine gets converted into 5-formyl cytosine, which can be converted into uracil, and this is read as thymine. Okay, so 5-hydroxymethyl cytosine, this gets converted into a 5-glucosyl hydroxymethyl cytosine, and this stays as it is, and this is read as cytosine in the sequencing methods. So what will be the result? Cytosine will be read as thymine, 5-methyl cytosine will be read as thymine, while 5-hydroxymethyl cytosine, it will be read as cytosine. So this is the TAB sequencing results. So let's compare it to the bisulfite sequencing and oxidative bisulfite sequencing. In the bisulfite sequencing, cytosine will be converted into uracil and will be read as thymine. Okay, in oxidative bisulfite sequencing also, cytosine will be converted into uracil and it will be read as thymine. The 5-hydroxymethyl cytosine, it can't be converted into anything, and that is why it is read as cytosine, but as well as 5-methyl cytosine is read as cytosine in bisulfite sequencing. But in oxidative bisulfite sequencing, 5-methyl cytosine is read as cytosine, but 5-hydroxymethyl cytosine, which get converted into 5-formyl cytosine, and it gets deaminated, and that is why it is read as thymine. Okay, so this is the final summary of all of the different procedures.